Hello everyone, this lab is about chromatography in part 1 and about color in part 2. So let me talk about chromatography first. Let's go to the pen tool. So chromatography, excuse the writing, actually means color writing, which is kind of silly. But actually what chromatography is it's a separation technique. And what it does is it takes advantage of the differences in interactions of mixtures with two phases. There's what's called a stationary phase. The stationary phase can be a piece of paper in this case, in the case we're doing, or it can be some solid glass beads, or it can be a gel. And then there's a mobile phase. And the mobile phase is usually a solvent. Oops, phase. And so typically what you do is you take your mixture and you put it, let's say sometimes we do this in a cylindrical column. And the column would be filled with, this would be the stationary phase. And then we put our mixture in here. So the mixture will be hearts, moons, stars, all the lucky charms. And then what happens is that these will dissolve in the mobile phase, and as they go through, they'll stick to the stationary phase, and because they're different substances, they'll stick to the stationary phase different amounts. And so if you have a whole series of test tubes here, and there's stuff dripping out of the test tube, out of the cylinder, as stuff comes out, right, things will come out at different times because things that stick to the stationary phase more will take longer to come through. Things that don't stick to this stationary phase will come through later. So maybe the heart comes through right away, maybe the star comes in here, and it's a separation technique. We can use it as an analytical, just to analyze, or we can use it in terms of, to actually separate things and get them into different test tubes. What you're going to do in this lab is you're going to do what's called paper chromatography. And in this case, in paper chromatography, the stationary phase is paper, and the mobile phase is a solvent. It's water, and I don't remember, water and ammonia. And by the way, the background to the lab manual will do a much better job than I do. It's got a lot of pretty pictures. So the idea is what you're going to do is you're going to take a beaker. That's a beaker. All right. But first, what you're going to do, let me erase some things here first. Let me erase everything. Let's erase this. There it goes. Bye-bye. Just like me erasing the board. So what you're going to do, I could hit clear all. We're going to use this guy. What you're going to do is you're going to take a piece of paper. Well, you're not going to do it. This is what we did. But you would have done. This is a really cool lab too. And you draw a pencil line on it. All right, this is just a piece of paper. And then what we would have you do is you take some M&Ms. M&Ms have dyes on them, food coloring. You put a drop of water on it and let the dye come into the water. And then you put a little spot here of the dye, just one drop. And then maybe this is a brown M&M. Maybe this is red M&M. Maybe this is green M&M. And maybe this is blue, I don't know. One I found online, a picture of it. All right, and then what happens is these dyes are frequently made from more than one thing. Then you're going to take this whole thing and you're going to put it in a beaker. Okay, again, you're not going to do this. And so here's my piece of paper. Right, it would sit on the bottom actually. And then I'll put some liquid in here, maybe up to here, some solvent. And here's my spots, right? Actually, you usually put it below the spots. right? And then what will happen is the solvent will move up by capillary action. And then when it's done, you'll see that these spots are going to break up into other things. And then the solvent, let's say the solvent moves to here, all right? And the solvent started here. So when it's done, you take your paper out, and you measure this distance. This is how far the solvent traveled. And then you'll measure each spot. So let's say this is the brown. I'm just going to do 2 of them, and we'll do the green. I'm not going to do all 4 of them. And let's say that what happened is that you had 3 spots. I'm just making it up. And here you had maybe 2 spots, like that. You can calculate a value, but you can see you've separated. So this is the original spot. Okay. The RF value is the distance that the spot travels over the distance that the solvent travels. Right? So for example, this would be right here. This would be how far that spot traveled. And then this spot here, that would be the distance that spot traveled. And you're going to calculate those. And so what we have online is data where I actually took a picture of some of this, and you can do the best you can to measure it. All right, so that's the separation. You're going to do it with we have 2 of them. We have one from just food coloring and one from M&Ms. All right, so let's do this again. Let's click the eraser and start erasing. Okay, the second part of the lab is about color, and you're going to measure the light absorbed. Well, again, you're not going to measure it, but we're going to talk about light absorbed. So first of all, anything that is colored, any solution that has color, that means it absorbs light in the visible region. Now, remember, you don't see the light that's absorbed. You see everything but. If the light's absorbed, it's gone. Where did it go? Well, it can move electrons up and down between layers where they live. It can make molecules spin around. It can do all sorts of things. It just depends on the wavelength and the frequency. And we talked about that in the lecture, and there's some of that in the background. What you're going to do, or what we did, is we're going to take food coloring and we're going to run what's called an absorbance spectrum. And in the absorbance spectrum, what we do, it's a graph of absorbance, how much light is absorbed, as a function of wavelength. And typically we'll get a curve that looks something like this. And so that would say whatever the wavelength is here, this substance absorbs light here. Sometimes you'll have a curve where you may have 2 different things. So we'll just go absorbance and wavelength. We'll use lambda for wavelength. Sometimes you may have something that looks like this. It absorbs there and absorbs there, and has 2 peaks. And these would be the wavelengths that it absorbs. And what you should find is that the wavelength that the light absorbs is not the same as the color. And the idea is that you'll develop the color wheel. And by the way we measure this, we use a device called a spectrophotometer. And it's a real bummer that you guys can't do this lab, because it's a very common piece of equipment. Every lab in the universe has a spectrophotometer, but all it does is it measures absorbed light. And on a video, I don't have time to go through how it works, but it's really easy to do. You put the thing in a compartment and you basically push go. But the idea that what you're going to try to do in the lab, or from the data, is you're going to get the color wheel. You may or may not have seen this. So the color wheel you just go red, orange, yellow, green, blue, and violet. That's supposed to be a G. And what this tells you is that if something looks red, it absorbs green. It goes opposite. If something is yellow to the eye, that means it's absorbing in the violet. If something is blue to the eye, it's absorbing orange. And what you should see is, say that the blue food coloring, hopefully the data will show that the blue food coloring absorbs orange light, right? And so what we've given you in the lab is I gave you a spectrum that's got a couple of different ones. So it's got like, I don't know, let's say that that's blue. And then another one that looks like this, that's the yellow. And you'll be able to see there's a very clear key. And then you can develop the color wheel. And then it asks you some questions about it. So pretty much that's what you're going to do in this lab. Again, you're not going to do it. You're going to work up the data. So what I've given you is the data, and then I've given you the data sheets from the lab manual that you can use the data to fill out, and then you can upload that. And then I gave you the questions as a separate document, and you upload those, and then of course the score will add up to 50, like always. All right, I'm available on Zoom during my normal times. If that time doesn't work and you want to shoot me an email, I can make a Zoom appointment for you because I'm not doing a hell of a lot right now. Actually, that's not true, but anyway. So that's for the chromatography experiment.